Morning to one and all gathered here. It is a pleasure that I welcome you all to this virtual webinar on recent advances in protein engineering with a focus on antibody drug discovery, organized as a part of Silver Jubilee Webinar series. Prayer is not asking, it is a longing of soul. It is a daily admission of one's weakness. It's better in prayer to have heart without words than words without heart. So let us begin this event by seeking the blessings of Almighty. I request Ms. Jayalakshmi of 2nd BSc Biochemistry to present the prayer song.
सत्यव्रतं सत्यपरं त्रुसत्यं सत्यस्य योनं निहितं च सत्ये सत्यस्य सत्यं ऋतसत्यनेत्रं सत्यात्मकं त्वां शरणं प्रपन्नाः वाणी गुणानुकथने श्रवणौ कथायां हस्तौ च कर्मसु मनस्तव पादयो नः स्मृत्यां शिरस्तव निवास जगत्प्रणामे दृष्टिस्तथां दर्शनेस्तु भवत्तनूनां नमो भगवते तस्मै कृष्णायाद्भुतकर्मणे रूपनामविधेयेन जगत्क्रीडधियो यतः.
I take this opportunity to invite our head of the department, Dr. P.T. Srinivasan, to address the gathering. Please, sir.
Thank you, Shivashankari. A very warm good afternoon to all the participants and heartful welcome to Mr. Vignesh Venkatraman from Agenus UK, Cambridge, and the participants from various city colleges in and around the city. A very warm good afternoon to one and all present here. So our department is celebrating the Silver Jubilee webinar this year. Having started in 1995, we ran through a lot of programs, starting with an undergrad program from BSc. Then we moved on to have the postgrad program, MSc in biochemistry. Then we have been converted to research department with the MPhil degree. So the department is slowly progressing and we have reached the first milestone of 25 years completion. So to mark this year, the department is organizing a lot of webinars, sections of the society in and around the world, and share their knowledge and expertise with the students, for the department students and the college students and whoever participates in this. So I am very grateful to all the team, especially the Neuberg crew team. The biochemistry club of our department is really doing a yeoman service of just organizing. They have become, although they are all science students, I'm really proud that they are all wonderful computer science experts. They are very good in information technology, and you name anything, ask them a video or YouTube streaming, Facebook streaming, whatever, anything. Even I feel even the computer science students won't be able to, an average student won't be able to perform so much. So I'm really proud of the team that is doing this wonderful work, and congratulations to the team and the organizing secretary for organizing this webinar. So before the start of the webinar, I've been just chatting with the speaker, and I already asked him a few questions and he was trying to answer all those questions. So this topic, the protein engineering, that is one of the key aspects that is going, it's a hot topic, I should say, of the present situation in the COVID, with our country's indigenously produced Covaxin and other vaccines of the COVID, Covishield, Pfizer and Sputnik. There are so many things that are coming up. In this scenario of a lot of vaccines coming up from different countries and different laboratories and research scientists, this topic, that is the protein engineering, becomes a very, very important area, especially for the students, like how protein engineering is helping being a biochemist, how the industry, the role of industry and the role of biochemist in the in protein engineering, and how it helps us in generating you can say a drug discovery, or you can say something related to vaccine. So the basic knowledge of this protein engineering and the antibody drug discovery, the topic of this today's webinar. So I'm eagerly awaiting, like most of you, to listen to the content of what's going to be there in the recent advances of this protein engineering. I once again congratulate the team, and I quoted in the previous webinar, I quote again in this webinar, when Winston Churchill said that the nation had the lion's heart, and I have the luck to give the roar. So I am lucky to give the roar. So with this, I just over to the MC team. Welcome you all once again.
Thank you, sir. I invite Ms. Lakshmi Sri, third BSc Biochemistry, to introduce the speaker of the day.
It's a deep privilege to introduce our dignified speaker of the day, Mr. Vignesh Venkatraman, a versatile and committed scientist with more than 14 years of experience in leading projects in the biologics area in multinational teams. Our speaker has expert knowledge across a wide area of early stage drug discovery, including phage display, protein engineering, molecular biology, screening, automation, and bioinformatics. Project management experience in multinational, cross-functional team and interactions in Europe and United States. Currently, senior scientist in Agenus UK Limited, Cambridge. Key achievements of speaker includes significant contribution to scientific leadership in the antibody discovery group. Led multiple phage display antibody discovery from early research to IND submission, some of which has led to successful collaborations with external partners like Gilead. Key expertise includes phage library constructions, protein engineering, molecular biology, high throughput screening assays like ELISA and alpha screening, SPR screening and kinetics, cell binding assays using flow cytometry and image devices at AstraZeneca, Cambridge, UK. Our speaker was co-inventor at anti-PSL arm bispecific antibody and currently in clinical trials for the prevention of Pseudomonas infections. Extensive experiences in monoclonal antibody drug discovery using phage display platform on successful projects. He is independently working in mammalian cell culture and purification of monoclonal antibodies that were isolated from phage display libraries, and he is also co-author for the scientific publication at leading journals. Mr. Venkatraman has attended key conferences and meetings held at Protein and Antibody Engineering Summit Europe, Barcelona, November 2021, and Next Gen Immuno-Oncology Congress, London, March 2019. Our speaker holds 2 patents on combination therapies using anti-Pseudomonas PSL and multi-specific anti-Pseudomonal PSL and PCR binding molecules and uses. He has also published research papers in prominent international journals, and his achievements are truly inspiring. Over to you, sir.
Thank you so much for the wonderful introduction. Just want to start off by with a huge congratulations to the Biochemistry Department for celebrating the Silver Jubilee, and to you, sir, Dr. Srinivasan. And again, it's fantastic that you're doing these webinars. I think this is really informative for the students, and I'm glad to be part of this webinar series. And also special thanks to Nagalakshmi ma'am, who was kind of like an inspiration factor for me personally as well. And yeah, thank you so much for the invitation, and I hope the talk today you find it somewhat useful going forward. So I'll start with my presentation. Okay, hopefully you can all see my screen. So today's presentation, like I was introduced, I'm going to be focusing on protein engineering and also how it's applied in antibody drug discovery. And this is kind of the area I've been working on, like I was introduced. Before I go into the details of the talk, I thought I would probably go through my journey, how I got here. I think that would probably be helpful in many ways. So I started off with my bachelor's in biochemistry. I did that at SRM in Arts and Science College in Chennai, and this is where I had the privilege of learning under passionate teachers like Nagalakshmi ma'am, who further inspired me and a few other friends actually to explore further. So one thing I wanted to highlight here is, during the second year of this undergrad, I did the workshop at a private company in Bangalore. So they were running this course in recombinant DNA technology and PCR. And back in the days when I was studying, the undergraduate was mostly, theoretical, and the practical aspects were there. But I wanted to learn a bit more about the things that I studied, practically. So got a few of my friends and I went to this course, and that really laid the road for the future, essentially. So there I spent 4 or 5 days doing DNA manipulation, basic, transformation, bacterial culture, gel electrophoresis, and things like that. That really helped me sort of give me reassurance. This is the area I want to sort of move forward in. And then in 2005, I moved to UK. I joined a university called University of Sussex. That's located in the southern part of the UK, Brighton, as shown here. Let me just get my laser pointer so now you can see. So there I was doing this course and master's in genetic manipulation. Unlike many master's courses in India, which is usually 2 years, most of the courses, the master courses in the UK, are usually 1 year. And it is heavily involved with research as well. The topics that you study, you have to do a lot of research. You have to read journals and publications and come up with your own interpretation of various research topics. And part of my master's, I was working, I think it was 9 months, I was working on a project. This is to do with manipulation of Bacillus thuringiensis. This is a bacteria that's used as a biofertilizer. So my supervisor at Sussex University, Dr. Neil Crickmore, he's an expert in that area, and this is what he was working on. And I worked in his lab for 9 months trying to sort of mutate a strain. And there I learned pretty much all the basics in molecular biology and DNA manipulation and everything. And once I finished this course successfully, I was lucky enough to find a job at a company called Genzyme. And I would say this is kind of the first time I got introduced to the antibody discovery field. And Genzyme, at that time, they had the sites in Cambridge focusing on antibody discovery. And I joined them. I was there for a year, learned a lot of screening technologies and basics of phage display. Some of this I'm going to actually go through today. And then after that, I joined Cambridge University. Cambridge University Oncology Department, Professor Ashok Venkatraman. So Professor Ashok is originally a medical doctor. He moved to London, University of London, I believe, many years ago, many decades ago, to do his PhD, and then he did his postdoc. And then he now is working as part of Cambridge University. He has his own group, and he's also the director of MRC Cancer Cell Unit in Cambridge. And his focus is around DNA damage and cancer, and looking at the BRCA2 genes and so on. So I was lucky enough to be in his lab. I mean, it was all surrounded by a lot of passionate scientists who would work around the clock. You can go there like 2 o'clock in the morning or 3 o'clock in the morning. Someone will be working, doing experiments, once or that. So I learned a lot working in that lab before I moved on to MedImmune. MedImmune is a company which is now being branded into AstraZeneca. So it was part of AstraZeneca when I joined there. And MedImmune MedImmune is also one of the companies that was pioneers of phage display. So I will talk about an antibody later on. So the first monoclonal antibody, fully human antibody that was ever approved was actually developed by scientists who actually worked in the early days of MedImmune, or it was called Cambridge Antibody Technology back then. So I was there for close to 6-7 years. I was working in the Antibody Discovery and Protein Engineering group, mainly doing phage display, and we also did something called phenotypic screening and technology development, where we improve various aspects of discovery technologies. And this is where I was privileged enough to be part of a team where we actually invented an antibody against Pseudomonas infections, and this is now in phase 2 clinical trials. So 5 years ago, just a little over 5 years ago, I joined Agenus. Agenus Cambridge arm of the company. So Agenus is an immuno-oncology company based in the US, in the east coast of the US, Lexington, near Boston. At Agenus, we try and develop medicines or drugs, you can say, that kind of help immune system to fight cancer. So one of the things that we do in Cambridge is Antibody Discovery, and that's the group I'm based part of, and I lead Antibody Discovery projects. And I'm also heavily involved in phage display libraries and so on. So I think that's basically just to give a bit of introduction about me, and I will move into the topic now. So, as I mentioned, protein engineering. Protein engineering is a wide area. It's actually used across different fields, and I thought I would probably start with some basic introduction. What is protein engineering? It's essentially the conception and production of unnatural polypeptides or proteins, often through DNA manipulation of proteins that are existing in nature. So, as you all know, proteins are the fundamental building blocks, and proteins are essentially made from DNA, and DNA is the precursor to protein in a way, and each DNA codon relates to an amino acid, which then forms the peptide or the protein chain. So by manipulating this DNA, you can change amino acids in a protein, thereby improving its function, and so on, which is called the protein engineering. So engineering the protein by manipulating the DNA is essentially what protein engineering is all about. And this is done manually, like I said, with DNA manipulation in the lab, or you can also do it using computational design. So there are softwares now where you can actually input the protein sequences, and then you can use computer modeling to look at residues that you can mutate and change for the desired effect. So I also thought I would just put this word cloud there. So this one gives you like lots of terminologies and techniques that people come across when they talk about protein engineering, and some of this might be familiar too as well. So yeah, protein engineering it's kind of like, is spread everywhere. So wherever people work with proteins, they would have engineered at some stage. So before I go into further detail into my presentation, I thought I would sort of quickly mention where protein engineering is utilized. For instance, you can say, food and agriculture industry, protein engineering is utilized. For instance, you might have heard about microbial proteases. For those of you who are into bodybuilding, who are taking supplements which contains enzymes that helps you to process those supplements better, and those contain proteases, which sometimes are engineered. And then I mentioned the biofertilizer earlier, the Bacillus thuringiensis. So these are insecticides, essentially. These are engineered insecticides, which are used these days. And you're all probably familiar with GM crops, the genetically modified food products. So those have advantage in several ways. And then if you move on to sort of other areas like environmental and medical field, there are sort of heavy research going on around using microbes to tackle pollutions. There you have protein engineering, and there are also research happening there now in terms of biofuels. And then if you talk about the medical field, in regenerative medicine, for instance, you have collagen-based polymers, biopolymers to make like skin grafts and things like that. So the protein engineering basically is everywhere. And then this is my area of expertise, obviously, drug discovery and development, and protein engineering is utilized across different areas. And, you can talk about cell therapy, and now we have different forms of cell therapies. For instance, CAR-Ts, where protein engineering is heavily involved. And in the area of we are living in a pandemic after all, and you're all familiar with vaccines. And protein engineering is a key part of vaccine production, for instance. And then we have antibodies at the end, which is what I'm going to talk about today. So let's just go through the past for a bit, and then try and sort of look at the history or, the story of modern medicine. So essentially, this starts in the 1700s, also in a similar situation to now, so where we had the smallpox problem, where a lot of people were dying because of smallpox, and there wasn't any effective treatment, or vaccination wasn't a thing before smallpox, for instance. And this is the time when a medical student, Edward Jenner, he was making his observations. So he was looking at his dairy farmers who were contracting something called the cowpox, which is a different form of disease, viral disease, and they were getting these blisters because of the cowpox. And he noticed that these farmers weren't actually getting the smallpox; they are somehow immune to smallpox. So he kind of thought about doing an experiment. So he took a 7-year-old boy, and then he scratched his skin, and then he took the fluid from this cowpox, and then he injected that into this scratch, and that immediately formed a blister of cowpox. And then once that healed, he took a fluid from a smallpox blister, and then he gave it to the same boy, but this time he was immune to smallpox. This is how kind of he theorized, okay, so you can, for instance, give similar viruses to give immunity to somebody, and they will then, become immune to the other virus. So this idea was further developed by other scientists, and that's how the smallpox vaccination came about. And that's kind of like the mother of all vaccinations, if you can say that. And then if you move on to sort of the 1800s, Louis Pasteur, who was a microbiologist, he was actually doing a lot of discovery at that time. He came across a little boy who was bitten by a dog. And at that time, rabies was a serious concern, and the mother was really concerned about this boy contracting rabies. And she took the boy to Louis Pasteur, who was researching on this. And he wasn't a medical doctor; he was just a scientist working on different things. One of the things was the rabies virus, and he kind of he has done some experiments in rabbits, and he thought, okay, we'll try that because of the plight of the mother. So worried about losing the boy to rabies. So he was convinced to give inoculations of rabies virus. And miraculously at that time, the boy recovered and he did not contract rabies in the end. So that kind of laid part to the rabies virus. And this is the first time somebody's actually used an attenuated virus as part of the vaccination strategy. And then in the 1900s, people were mostly using plant extracts and bark extracts and using this as medicines. And a few of them are mentioned here. And aspirin is something we still use today. This is a known analgesic or antipyretic, for instance. This was originally from willow tree, and this was used to treat fever. And these are all like happening when there wasn't major breakthrough in terms of biotechnology or the techniques and things like that. But the real breakthrough, if you can say, happened when Alexander Fleming discovered penicillin. And there's another story attached to this. So Alexander Fleming was a scientist who was actually working in London. He was doing a research on a pathogenic bacteria, Staphylococcus aureus. And he was growing this bacteria in a petri dish, and he went on a holiday. And when he came back from holiday, he realized his petri dishes, he didn't actually protect them very well. And then there was something moldy growing on those petri dishes. And then when he looked at them under the microscope, he realized these are that there is something happening there, which is kind of stopping the Staphylococcus to grow effectively. And then later on, long story short, he discovered penicillin being the active ingredient that's let out by those molds, which was basically stopping the bacteria to grow. And that's how penicillin was discovered. And penicillin, as you know, is one of the effective antibiotics, which has helped, back in the day. I think this was just close to the Second World War. And a lot of soldiers, injured soldiers, were saved because of penicillin. And even till today, I think penicillin is one of the major antibiotics used by hospitals and doctors around the world. And if we slightly move towards the later part of modern medicine, so 1973, you had the first atomic resolution structure of the antibody published. And then in 1975, Kohler and Milstein invented hybridoma technology. So hybridoma technology is really the first time where you were able to produce antibodies in slightly larger scale, monoclonal antibodies, I should say, and they were awarded a Nobel Prize for this as well. And then in 1985, phage display technology was invented. I will go through phage display technology today. And obviously, when they invented it for the first time, it was quite basic, and there's been a lot of improvement. And phage display technology is one of the key technology people use these days in the development of therapeutic antibodies across the world. And in 1986, the first monoclonal antibody was approved by the Food and Drug Administration Agency, which is the regulatory authority in the US, FDA. And a few years later, 16 years later, I think, in 2002, first fully human monoclonal antibody was approved. And this is the antibody that was originally discovered and developed by the group of people I worked with at MedImmune, when it was Cambridge Antibody Technology. And then in 2005, FDA approved the first bispecific antibody. So when you talk about antibody, you usually have specificity toward one antigen. In this case, this is the first time the first antibody was approved where it actually targets two different antigens at the same time using one molecule. And then 29 years later, since the first monoclonal antibody was approved in 2015, the 50th monoclonal antibody was approved, which you can imagine it's taken 29 years actually to have the 50th molecule to be approved. And this kind of shows like how the technology has evolved over the years. And then another milestone I want to highlight here, because we're talking about protein engineering, 2017 is for the first time a CAR T, which is a cell therapy, was approved by FDA. And this is basically, you know, where they take the patient's T cells and they engineer the T cells to have a chimeric antigen receptor displayed, and then they put the T cells back into the patient to treat a form of cancer. And this is a major milestone in terms of for the cancer patients, essentially. And then this year, in fact, a couple of months earlier, I think in May 2021, the 100th monoclonal antibody was approved by FDA. And if you think about it, you know, it's taken 29 years to develop 50 antibodies, but then in 7 years or in 6 years, for instance, we've managed to get another 50 antibodies approved. It really speaks about the advancements and understanding in antibody drug discovery. Obviously, there are other drugs and different sort of developments happen in modern medicine, but because of the theme of the day, which is antibody drug discovery, we have focused on just the antibodies here. So talking about antibodies, I want to sort of touch upon immune response. So this is something I think you will all be familiar with. You know, we've all learned about the innate immune system, the innate immunity and the adaptive immunity. So the innate immunity being your first line of defense, that is your skin, mucus, hair, and other epithelial surfaces. For instance, sneezing or coughing to eject microorganisms or germs is a way of way your body is trying to protect yourself from other pathogens. And once these pathogens breach the first line of defense, that is the skin and mucus, it enters the bloodstream. There are a wide variety of white blood cells or leukocytes that patrol the bloodstream, and they are ready to respond to infections. And you would have heard about phagocytosis, which is one way that's taken care of. And also there are cells which play active role in innate immune response as well as adaptive immunity. So if you think about adaptive immunity, this is where the antibodies comes from. So you have the B cells or the B lymphocytes, which are white blood cells that produce antibodies. So when a foreign antigen, or for instance, virus or bacteria, they enter your bloodstream, when the receptor binds to the B cell surface, it stimulates the B cells to divide and mature into identical cells. And this is called a clone. And then the mature B cells essentially secrete millions of antibodies into the bloodstream and also to the lymphatic system. So one of the major advantages of this adaptive immunity is the immune memory. So somebody who's contracted a virus, for instance, chickenpox is a classic example. So many of us would have got chickenpox when we were young, and you would have your immune system would have been introduced to the virus, and that kind of creates an immune memory. So even if you come across the same virus years later, these immune memory would kick in and then produce more antibodies against the virus and tackle it. And this kind of theory, works in the COVID vaccine as well. So the vaccine contains part of the virus protein, and then once your body is introduced to that particle, your body produces back antibody against the viral protein. And then when you come across the real virus, so your immune memory essentially, takes care of the virus and protects you from serious infection. So I mentioned about the antibodies. So I want to tell you, if your body is totally able to sort of produce antibodies, why do we need to manufacture antibodies in the lab? So I want to sort of give you an example. One of many reasons why we need to sort of engineer antibodies against disease-specific antigens. So here I'm going to talk about the immune checkpoint inhibitors. So immune checkpoints are proteins that are expressed on normal cells that helps to prevent the immune system from attacking your own cells. So I've got a little video here. Let's hope it works. So you have the T cells usually, which role is to basically patrol the immune system and then attack any sort of danger. So this basically goes and binds to cells, and you saw the cancer cell, it was detected by the T cell, and it destroyed the cancer cell. And that's the normal situation. And then when it goes and binds to the normal cell, there are receptors on the normal cell which sends an off signal, and the normal cell get not attacked by the T cell. But then the cancer cells take advantage of this mechanism, and they also produce the receptor, and then they pretend to be like the normal cell. And this is called the immune checkpoint escape, for instance. And one such protein is called the PD-L1, which is expressed on this tumor cell. So when the T cell tries and binds the tumor cell, and the PD-L1 signals and says, okay, I'm just a normal cell, and then the immune response basically shuts down, and the tumor cell escapes immunity. So what we're trying to do is, I don't know if you saw the video, there was this example. Let me just see if I can go back and play the video again. So you have the T cell, and in ideal situation, it binds the tumor cell, and then it detects it's dangerous one, it can kills the cells, and then it binds a normal cell, and then you can see the signaling there, and it basically, doesn't attack the normal cell. And this plus sign here in red is kind of like the signaling one. And then the cancer cell expresses this signaling receptor, and that turns off the immune response, and the tumor cell and T cell actually lets the tumor cell alone. So what we're trying to do is, using the immune checkpoint inhibitors, we try and inhibit this signaling, thereby making the tumor vulnerable to the immune system. And that's kind of what we try to do with the antibodies. So we try and create antibodies against these targets, thereby switching off this immune checkpoint, essentially. So antibody discovery methodology. So there are many ways antibodies are discovered these days, and I've just put a few of them here. And the one on the very left is essentially called immunization. So you have animals such as rabbits, horse, sheep, mouse, for instance, which are immunized with the antigens of interest. And then the plasma, or the blood cells are harvested, and then through a lengthy process, you in the end get like a polyclonal mix of antibodies, which means you have different antibodies produced against this antigen produced by different B cells, for instance, and then it's a polyclonal mix of antibodies. And these are mainly used for research purposes when you talk about ELISAs and other flow cytometry and things like that. So we use polyclonal antibodies. And then we have this hybridoma technology, which I mentioned earlier. So this is, again, another interesting way of generating antibodies. So where you immunize the animals, and then you use a tumor cell. You fuse the B cells from the immunized animals with the tumor cells and form like an immortalized cell, which then continuously produces antibodies And these are usually, you can have monoclonal antibodies using this process. But then the drawback here is, again, it is dependent on the species that are immunized against the antigens. So in this case, if it is mouse, you can only have mouse antibodies, which you can't use for therapeutic purposes, obviously, because the human body would react to mouse antibodies as being a foreign object. And then there are, latest advances in other sort of transgenic animals. These are animals which are genetically modified to more human-like, for instance. So similar principles, you can immunize them with antigen, and then through a lengthy process, you manage to sort of get humanized antibodies or human antibodies because they are humanized animals. And then what I'm going to talk about today are the display technologies. I'm going to focus on phage display, but there are other display technologies out there as well. So for instance, we have ribosome display, where antibodies or antibody fragments are displayed on ribosomes. And there are mammalian display technology where, again, you can display a full antibody, full-length antibody on B cells, and then you can have a library of these, and you can basically have your target antigen bind to this library, and then select the best antibody out of the whole lot as well. But today I want to focus on phage display technology, and that's the field that I'm working in. But before that, I want to sort of introduce the antibody, the structure of antibody. And I'm sure, you would all learn about antibody structure if you've not already learned about it. So it's the Y-shaped molecule. And what you have here is, so you have the heavy chain domain in blue, and then you have the light chain in green. And then in the heavy chain, you have this part, which is the slightly lighter color, the lighter shade of blue, which is the variable region. And then you have the constant domain, which forms the Fc of the antibody. And then within the variable region of the heavy chain and light chain, what you have is called the complementary determining region, or the CDRs, which are hypervariable domains. And these are the ones that are responsible for binding to the antigen. So within the CDRs, so you have, like, in each of the heavy chain and light chain variable region, you have 3 CDRs. Like, if you look at the structure, they look a bit like this. And you have the loops, for instance, formed by these CDRs, CDR 1, 2, and 3. And CDR 3 of the heavy chain and CDR 3 of the light chain are essentially known to be playing a major factor in antigen binding. Since this is like a conventional antibody, so you have 2 antigen binding sites as shown here, and as a monospecific antibody, these are specific against one type of antigen. And coming to phage display technology, so you would have all heard about bacteriophage, which is a form of virus. And you would have all heard about TMV virus, which is another kind of bacteriophage. And what we use in phage display is this filamentous phage called M13 bacteriophage. And that's what it looks like if you look under an electron microscopy. And it's actually quite small. You can't really see it. Even using a normal microscope, you need really an electron microscope. And that's the size of the viral particle, for instance. And the nature of this virus is essentially it contains a single-strand DNA as the genetic material, and then which is surrounded by a gene A protein, which is a code protein, and then you have the gene 3 protein, which is beneficial for us. You will see about that in a little while. And basically, what it does is this virus is capable of infecting bacteria. It uses the bacterial machinery for its own advantage. So what it does is it binds to the F. pili of the E. coli. E. coli is a strain of bacteria. And then once it enters the bacteria, it unpackages the single-strand DNA, and it uses the bacterial machinery to basically multiply its own DNA and then produce a protein. So there are, using the system, it produces new single-strand DNA circles, and then all the viral proteins required to make the new virus are all produced and then packaged. And then basically at the end, you get several of those bacteriophage made using the cell. And the clever thing is the M13 bacteriophage is able to do this without killing the bacteria. And in each cycle, in each bacterial cycle, in which case, in E. coli, the bacteria doubles every 20 minutes. For instance, if you have one bacterial cell, in 20 minutes, you'll have two. In another 20 minutes, you have 4, 8, and so on. So in each of those cycles, you're able to have a 300 phage produced approximately. And this gets executed from the cell into the media. So what we do in phage display technology is, we take this bacteriophage, and I mentioned in the antibody, we have this variable region, which is responsible for the antigen binding site. And what we can do is, if you look at the antibody genes, it looks somewhat like this. You have the variable range followed by the constant domains. And same for the light chain as well. Light chain, you have only one constant domain followed by, before the variable region. So if you take these two variable genes and then put them together, you can have a linker. Linker is like a small peptide fragment which kind of connects the two chains, two variable chains. And that would look like this. It's called a single chain variable fragment, or scFv. It's about 25 kilodaltons in size. And this kind of has the same properties in terms of the binding properties of an antibody, because you got the variable part there containing the CDR regions which bind the antigen. So using sort of this gene, we can then clone that into the phage display vector. So vector is basically an engineered plasmid which forms the single-strand DNA I showed earlier inside the bacteriophage. And as I mentioned, we have this coat protein, we have this gene 3 protein at the top. So this bacteria, this plasmid, you can actually engineer the single chain FV gene along with the gene 3 protein of the bacteriophage. So once you then insert it into the bacteriophage, when the gene 3 protein is expressed or displayed on the surface, your scFv, or the antibody variable fragment, is also displayed. And this is just one clone. I'm just showing as an example, like one clone, how it's done. And you can do the same thing, for instance, by taking 1 million different antibody fragments, and then you can clone them all into bacteriophage and then have a library of bacteriophage displaying 1 million different antibodies, and this is called an antibody library. And the key thing here is the phenotype to genotype linkage. So you can, for instance, use this bacteriophage displaying your antibody fragments to bind your target antigen. The key thing is, because the genetic material is encoded inside the bacteriophage, you can deconvolute the antibody or the sequence of the antibody by sequencing the phage bit, essentially. And that's the phenotype and genotype linkage. So this is like a quick sort of overview of how the phage library or the scFv library is constructed. Like I said, so this is an antibody library, right? So we tend to create these libraries by isolating blood from healthy donors. And what we are specifically interested in are the lymphocytes. Like I said earlier, B cells are the ones produce antibodies. So they contain the genes that are encoding these antibodies. And basically, so we take those lymphocytes from non-immunized donors, and then we extract the RNA from these patients, from these cells. From there, you can synthesize the cDNA by reverse transcription. And then we can use germline specific primers to PCR amplify, which is the PCR is polymerase chain reaction. You can amplify specific genes, for instance, here, the heavy chain and the light chain variable genes. And these are assembled as an scFv I showed in the previous slide, which you can then engineer into the plasmids. And then you basically clone that into the bacteriophage. And as I mentioned earlier, using the process, you can create antibody libraries. These are often, really, really large in size, and that kind of in a way represents what your immune system would be able to produce, for instance. And this is kind of like the feature of the phage display library. This is, like I mentioned, the phenotype and genotype linkage. And you can say this is kind of a replica of your adaptive immune system, particularly if you talk about the antibody diversity, because we are engineering the genes from human blood cells. into the library, so you can, in a way, create the antibody diversity that you would naturally see in human body. And the typical size of a library is about 1 times 10 to the 10 to the 10. Essentially, if you think about in crores, it's about 1,000 crores. That's how many different clones you would have in a typical library. And the major advantage of phage library is, because of the size of phage particle, you can have quite a large number of phage particle in a very, very small volume. And here I took a picture of an Eppendorf tube, we call it. This is a microtube. It's only 4 centimeter in size. And there I have about, I think this is about less than half a mil. In fact, 400 microliters of phage library in this tube. And that basically has, you can imagine, 4 times as the volume I've shown here on the last bullet point here. So now we have the phage library. The next thing is to make the proteins that you want to select against. So like I mentioned, earlier when I talked about the T cell, how cancer cells can evade immune system, I mentioned, tumor cells are able to produce receptors or proteins on the cell surface. Our aim using the phage display library is to find binders against those proteins. So one way to do is actually engineer those proteins recombinantly. So here I'm giving an example. So you can get the sequences of these proteins from various databases and literature. And if you're studying bioinformatics, you would come across a database called UniProt. UniProt is somewhere where you can find lots of different proteins, millions of proteins, in fact, across different species. And I've shown here an example of what you will get. This is like a PD-1 antigen. So the sequence of the antigen looks like that. It's a protein sequence that you get. What you can then do is reverse translate to get the DNA from the protein. You can use tools for this, for instance. And then these, the DNA sequence is codon optimized. So because, we're going to use a recombinant, transient expression system, you need to make sure that the codon selection is compatible with the expression vehicle, essentially. So if you're using bacteria to produce this protein, then the bacteria prefers certain codon usage. Codon as in, the AGTC, the different codons that you have for each amino acid. So there are software tools now you can use to automatically codon optimize the DNA. So in this case, I'm talking about mammalian expression, and that's kind of what we do when you talk about human antigens, because for the purpose of producing antigens for therapeutic antibody discovery, you need to make sure that the post-translational modification is correct, and other things like glycosylation and things like that should be perfect. For that, you need to be able to produce that in a mammalian system, and that's what we tend to do. So what we do is basically the DNA. So now we have the DNA, which is then codon optimized to get the exact DNA that you want, which are then engineered into an expression vector. And there are, several vectors which have different components to it. You will see some of this mentioned here. So once you have cloned the gene of interest onto this vector, you can then transfect them into the mammalian cells. And the common ones people use these days are CHO cells, or for instance, Chinese hamster ovarian cells, or you can also use embryonic kidney cells or HEK cells, human embryonic kidney cells or HEK cells. And then what happens is, so the DNA, we typically conjugate it or couple it with a lipid sort of material, which is a transfection reagent, which then takes that inside the cells, and it basically, the cell machinery takes care of producing these proteins, and which are then released into the supernatant. And then you can use the cell culture to purify these proteins using various methods. Some of them are mentioned here, which is the affinity chromatography. And in affinity chromatography, you can automate everything fully. And what we have here is a column which is packed with something called a protein A resin, which binds the antigen using a tag attached to the antigen. And then using affinity chromatography and other methods, you can purify the protein. which you can then do a QC by SDS Phage, for instance, polyacrylamide gel electrophoresis, and typically, ELISA or something like that would be done to make sure the protein is intact and usable for the purpose. So now we have the library, and now we have the antigen. So how the phage display selections are done. So as I mentioned, the phage display library contains millions of different antibodies displayed on the surface of bacteriophage. And then you have the recombinant antigens we made on the previous slide. So what we then do is we come up with two methods. So one of the common method is called the panning selection or solid selection, also known as. So where we coat the antigens on a surface of a plastic. This could be a 96 well plate or like a little test tube, which contains special coating where the antigens can stick to. And then once you have the antigen coated on the plastic, you come in with the library, you throw the library on the antigen. And what happens is some of the proteins, some of the antibody fragments displayed on the phage would bind the antigen, and some of them won't. What we do is we use several saline solutions and detergents to wash these non-specific or unspecific binders. And then the ones that are specifically binding the antigen are eluted using several reagents, which will detach the phage from the antigen. So you can then, as I mentioned, this phage contains the genetic materials. You can use the phage to infect the E. coli, and then the genetic material will be used to again produce the phage again. So the other method is soluble method. So as I mentioned, in panning method, you coat the antigen on plastic. Not all antigens would be happy to be coated on a plastic, so you need to keep them in solution for them to be intact. So we have the soluble method where we use different things called a biotinylation method, where you add a tag to the antigen. Basically what we do is, we use something called the microbeads, which are magnetic microbeads, which has something called a streptavidin, which binds the tag, the biotin tag. And then you can use this to pull out the interesting ones. And in the end, you have the eluted phage. You again infect the E. coli and recover them. And typically, this process is repeated for 3 rounds. So you have the library, you throw them on the antigen, you wash the non-specific ones, and then you take away the binders, and then basically you recover the phage and you repeat the whole process again. So you do that 3 times. After each round, you start with for instance, at the first round, you start with the library that's a size of 10 to the 10, and which contains, lots of different antibodies. Most of them won't bind your antigen. And then as you go through these selection rounds, so you start to lose all the non-binders, and then as you recover the binders, so you have all the clones that are binding get enriched, as in they multiply and they dominate your output essentially. And then what we do is we then take these phage and do a lot of screening. So depending on the target antigen of interest, we do different screens. One of the main thing is obviously to make sure that you can still bind the antigen. So ELISA is one of the traditional methods that we test these antigens, antibodies for binding. And then obviously until this point, the antibody fragment's still not a full antibody. It's still like this single chain FV displayed on a phage. So we can then engineer, so we'll have a handful of binders, which we can then engineer to make it into a full antibody, and that's the last stage. And then once the antibody is made, you can go again and do lots of different experiments to validate that is the right antibody that you want to have, and you can actually have the desired effect or function with that antibody. So I mentioned the antibodies, how they are made, how they are discovered, for instance. I want to sort of talk through like different formats of antibodies that are currently in research, and also some of them are approved for therapeutic usage. So I mentioned the native antibody, which is this Y-shaped protein, which has 2 antigen binding sites. This is a monoclonal antibody, in fact, monospecific antibody, because they bind the same protein on both arms. So I mentioned the bispecific antibody. So again, this is engineered antibody, and I don't want to go too much into detail, but he has other engineering done in FC region of the antibody that makes it easy to manufacture and and form the right bispecific nature. And as I said, this antibody actually binds two different antigens at the same time. So essentially, you can have two different functions with a single molecule. And again, likewise, you can engineer antibodies to have multi-specific antibodies. In this case, you have the antibody, the bispecific antibody, which at the C-terminus also engineered with two different scFv fragments. And here you can technically, bind four different antigens, for instance, hence it's called multi-specific antibody. And likewise, these are called bispecific T-cell engagers, so where you have two different single-chain FCs, FVs that are connected using a protein linker. And these are typically used as a T-cell engagers, meaning one arm of the molecules, for instance, this scFv will bind the T-cell, the other scFv will bind the cancer cell. And by this, they will bring the T-cells close to the tumor cell, and through the signaling, they use the T-cell to attack the tumor cell. And another one is antibody drug conjugate. And this is another interesting one where they take the normal antibody and they engineer to have some cytotoxic drugs on the surface of the antibody. And what they can do is they can use the binding arm of the antibody to localize to the, to basically take it to the specific location in the body. For instance, if you're talking about tumor, you can make sure that the antibody is taken inside the tumor, and then the toxic, the cytotoxic effect happens with the toxin that's the warlord, I think they call it. They then delivered to the site of interest, and then it's not affecting normal cells. It's only the tumor cells that are targeted by the toxins carried by the antibody. And then there's other formats as well, which I won't go too much into detail. So this is kind of how antibodies are engineered these days. And also, I think finally, I would probably give you an overview of how drug development happens. I said, in the introduction, when you talked about the history of drug development, I mentioned, it takes 29 years to make 50 molecules to approve. And then in the last 6 years, we had another 50 antibodies approved. It might sound like, it's a simple process, but actually it's very complicated than that. A typical drug usually takes about 12 years, and it's very expensive process. They say it takes about $2.6 billion to reach the market and patients. And also, the success rate is not that huge. Many drugs fail at different phases of drug discovery. And this is kind of like an overview of how drug discovery happens. So you have the target discovery phase initially, where you identify a target, which could be a protein expressed on a disease cell, for instance. And then we need to make sure that the target is druggable. Whereas, if you generate an antibody against a target, you don't want to have the same target expressed on a normal cell. So when you give the antibody to the patient, it can have non-specific effects. So you don't want to have any of that. So there is a lot of effort put into the target discovery and target validation phase, where you actually make sure that this is the right target to go after. And then after that comes the lead generation, where we use phage display technology, for instance, and other methods to identify leads against the target of interest. And then there's a lot of other activities happen during this phase. Whereas, we do the PK/PD, the pharmacokinetics and pharmacodynamics, where you test the antibodies or, the drug to see what the drug does to the body. Or, for instance, you put it through animals, and then how long it takes for the animals to clear the antibody. And then in PD, pharmacodynamics, you see what effect the antibody has on the body, for instance. So you do all of those things quite early on. And then once you have your lead candidate, you go through an optimization phase. Optimization as in, how we can make this molecule better, in terms of stability, in terms of its affinity or efficacy. So there's a lot of things happen there as well. So there, you basically have one molecule or, your lead molecule. You then try and make it better. And then you enter the preclinical development phase, where you do a lot of animal studies to understand the efficacy of your molecule in sort of in vivo setting, and before you move on to sort of clinical trials. And once you've gone through all these phases and you've shown that the drug is efficacious and is safe in animal settings, you then submit for investigational new drug approval, which is like an approval you have to get. For instance, in the UK and mainly in the US, you have the FDA, who look at your application, look at your molecules, and all the experiments you've done to make sure this drug is safe, and you can actually inject that into humans for clinical trials. And then clinical trials is the large phase where it goes through quite a lot of scrutiny. To start with, healthy individuals are given the drug to make sure it doesn't cause any harm to healthy individuals. And that's the phase 1. And then phase 2 and 3, where you have the patients where you show that the drug has an effect, and based on the results, you then try and submit your application for an approval. And so it takes a long time and a lot of scrutiny to produce a drug in the end. And then the antibody or drug, for that matter, can fail at any stage. And so that's kind of to give you an overview of drug discovery. And I think that's my last slide as well.
Thank you, sir. It was a very informative session. Dear friends, you can make this session more interesting and interactive by asking questions.
Sir, how do you get the idea of taking protein engineering for this webinar, sir?
How I got the idea?
Yes.
It's quite simple, really. So Nagalakshmi ma'am, like I said, was a major factor for me to be in this field. To be honest, she in fact she taught me the basic structure of protein when I was doing my undergraduates at SRM College. And thereon, obviously, I've been, doing various things, and this is the field I'm working on. So when she contacted me a few weeks ago, asking if I would be interested to sort of give a talk, I thought straight away, I'll be, very happy to do it. And this is the field I work on. So any sort of inspiration, I guess, I can give to the students. I think, this is my way of giving back, I guess. So yeah, so that's it. That's the reason, really.
Thank you, sir.
Hi, sir.
Hi.
This is Neuberg from second BSc, sir. I have some questions, like inspired of your journey through. So I have some questions apart from the topics, sir. Shall I,
Sure,
sir?
yeah. Of course, yeah. Go ahead.
Sir, I saw your journey. You have said that after your BSc, you have immediately gone to abroad for doing your MSc, sir. How was that possible, sir? Like, there's no small period of span time you were here. Moving immediately, how was it, sir?
Like I said, back in the day, so when I did my bachelor's degree, I was quite interested in sort of studying further in terms of biotechnology area. And I was looking at different options. And one of the options is to pursue a master's in India. And I've had cousins, who've gone abroad and they've actually done education, and they are working in different fields as well. So that kind of, I took inspiration from them as well. But the main thing I think is, the course I did during my second year, I think it was at Bangalore Genie, doing the recombinant DNA technology, the workshop, the 4-day workshop, that really kind of inspired me. Sort of, you read about all of these, for instance, in bachelor's, I read in the first year DNA replication, transcription, translation, and all that in very detail. But you can only visualize so much back then. 20 years ago, the internet wasn't the same. So we did not have smartphones. So for the internet, you have to rely on dial-up internet. So we don't have YouTube, where you can actually go and watch a video to understand the intricacies of all these mechanisms. So for me personally, attending the course where I practically managed to do all those things, that really inspired me to sort of, this is the thing I want to do next. And I was looking at different universities. And at that time, I think Sussex University came to Chennai, I think through British Council. They had this day where some of the, I can't remember, the staff members of Sussex University were there, and I had some interactions with them, and they introduced to me the course. And yeah, so it all went from there, really. I think, yeah, if you're someone who's looking to study abroad, my advice would be like, to make sure you know what you're really interested in, because when I came here, even though I've done all the course and I knew what I was coming into, it was still such an eye-opener, because the way masters or any matter of that matter education is done in the UK is completely different to India. So you're expected to work independently right from the start, and you don't really have lecturers or professors spoon feeding you. Basically, they will introduce the basics, but then they will leave it to you to go back and do your own research, reading journals and publications and understanding the concepts, etc. But they'll be there to guide you, but they won't actually hold your hand to take you through all of these. So that was kind of like an eye-opener for me. But I think I had the sound basics thanks to my passionate teachers like Nagalakshmi ma'am, who made it easy for me, really.
It was really inspiring, sir. I got inspired from your sayings as you got few years back. So one more question too, sir. According to your point of view, which country do you think it suits good for an life science aspirant, sir?
As in research or education?
Research, sir. Absolutely. Both. Like doing MSc and then further in research fields.
Again, UK, obviously I'll be biased because I'm based here. So UK is one of the leading countries in research, and so is US as well. And there are countries like Germany is also quite big. But if you talk about Europe, I would say Cambridge is now kind of like the biotech hub. It's one of the major hubs for Europe. And in the US, you have Boston in the East Coast, and then you have California is also quite big for biotech. And all of these places have universities and institutions attached to them, and industry. For instance, Cambridge, you have Cambridge University. There are other universities in Cambridge as well. And again, across UK as well, there are many universities, leading universities. And Cambridge is one of the biotechnology hub. You have major companies like AstraZeneca who are based in Cambridge as well. So yeah, it's very hard to say. But again, it all depends on exactly what field you're talking about. In general, I would say biotech, UK and Germany in Europe. I'm not saying other countries aren't good, but definitely UK and Germany are right at the top in terms of Europe.
Okay, so definitely there's a huge difference studying masters in India and doing masters in abroad. Am I right, sir?
That's correct, yeah. And I think also like some of the students I went to university with, they'd already done a master's in India, and then they came to do a second master's, for instance, in the UK, and they found it quite beneficial for them because it really prepares you as well. So yeah, that's just another way to look at it. Again, one thing I can't really comment is I did my bachelor's 20 years ago in 2002 to 2005. And I don't know how the education is now in India, so I can't really say whether now it's improved or it's the same. So it may be in some colleges or institutions it might be on par with many international universities. I know, like for instance, many universities, including I think there's some research happening at your institution as well, and other institutions across India and. Yeah, it's a difficult one for me to say. Yeah, but the way I think the culture is different. The way masters is approach is different. So you're expected to be more independent in the UK. That's one thing I can say. Yeah.
Thank you so much, sir, for spending your time and questions to be answered apart from the topic you covered, sir. Thank you, sir.
It's my pleasure.
Good afternoon, sir. This is Satya from 2nd MSc. First of
Hi.
all, thank you for your presentation. It was really very inspiring and interesting. I have a question about vaccines and antibodies. Are these both same or different from each other? And what are the difference between vaccines and antibodies?
I'll try my best to explain. So vaccines are usually, so you have two forms of vaccines, right? So you have the attenuated viruses. So for instance, we were all introduced to polio vaccines since childhood. It's a big thing in India for many years. So there you have attenuated viruses, means, inactivated virus. So that's given to you in terms of vaccination, where your body then produces antibodies against. Antibodies, one of the key immune responses against foreign particles, in this case virus. So it's the body that produces vaccines, or body that produces antibodies against the vaccines, which is attenuated virus. And then in terms of COVID, we have different forms of vaccines. For instance, the AstraZeneca one has like the gene that corresponds to the protein of the COVID virus is encoded into an adenovirus. It's a different kind of virus. It's a chimpanzee virus. So we code the DNA of that into this virus, and that's basically given as form of the vaccine. And basically your body reacts to that and produces antibodies. The main difference between vaccines and antibody is vaccines are usually attenuated forms of virus or viral particles, and then antibodies are something produced by your body.
Thank you, sir.
Good afternoon, sir.
Hi, good afternoon.
Yeah, my doubt is related to protein engineering. Is it possible to isolate a histag protein which is having a high prevalence of non-polar residues?
That's a specific question, so I need to know more detail about that to answer that question. But in general, I mean, histag is something we use routinely. So for producing recombinant antigen, histag is the common tag that we use, and then we use nickel resin to purify the histag proteins. But in terms of what you are having, is probably, I think I need to know more detail to answer that question, unfortunately.
Yes, sir. Thank you.
Excuse me, sir. Some questions are posted in YouTube. Shall I read it for you?
Of course, yeah.
Is there any challenges for protein engineering due to folding of proteins by Abhinami S?
Absolutely. I think protein protein folding is one of the major concerns. Obviously, it depends on the protein as well. So when we do produce recombinant proteins, we do a lot of QC. And for instance, STS Phage is a really basic one. And then we do something called ASEC, size exclusion chromatography, which also looks at the integrity of the protein. So yeah, protein folding is an important one that we look at. And because we do, I mean, we do protein engineering and protein expression all the time, this has kind of become like a routine thing for us. But certainly, protein folding is one key aspect. And we also look at, PTMs, when you talk about antibodies, for instance, antibodies can contain different motifs that can cause different problems, down the line in terms of formulation, manufacturability, stability, and so on. So there, PTMs are different forms of issues we come across as well, which we tackle.
Okay, sir. There is another question from Manjula Rajendran. How would you relate your topic to any speedy remedy, say for virus?
In what way? So there are companies in Cambridge, in fact, who are working on generating or developing antibodies against the COVID virus. And I mean, I think it's just, it is, if you think about it from the research perspective, producing an antibody against virus is similar to producing antibodies against any antigen. So we're not going to produce against the whole virus. It's going to be against the protein or some particle, some materials that's expressed on the surface of the virus. So, but then, like I said in the last slide, the antibody or, in general, the drug discovery development process, it's not an easy process. So typically, it takes quite a long time to go through all the various stages and development. And that's the one interesting thing. The COVID vaccine was actually generated quite quickly, and there were some concerns. People, usually it takes a long time to produce such thing, and now we have a situation where we managed to produce this in a very short time. And if you listen to the researchers, I think there's some nice videos of interviews or even documentaries made with the Oxford University researchers who were involved in producing the first COVID vaccine. they will tell you, you know, how they were working frantically. So when the first COVID was discovered essentially in China, some research groups in China, they published the genome sequence, and the Oxford University researchers, they got hold of the sequences over a weekend, and somebody, I think one of the lead researchers, continuously worked over the weekend and immediately got the research going, basically. And once before the whole pandemic situation happened, they already done a lot of work to produce vaccines against this. Like the early part of the vaccine development was already done. And then once we know that we are in a real pandemic, and it's not just the health that was affected, like a lot of people were dying, which is, you know, really, really sad, obviously, because no help could be offered for them. And hospitals and other health institutions were overwhelmed. And this also put a large, huge strain on other aspects. For instance, cancer patients and other patients who needed urgent medical care couldn't get to the treatment centers or hospitals because, you know, this was full of COVID patients. So this really, I think, you know, impacted in many ways. And also economically, I think many governments suffered because, you know, people weren't able to go to work. So there's a lot of factors, you know, that goes with pandemic. So there was like an urgent need. So if you don't actually come up with the vaccination really quickly, you know, it's going to have a serious impact globally. I think that's mainly, you know, was the driving factor. And you can think about it, you know, if one person can do a job and it takes him 10 days to do, and if 10 people join together, they can do the job in 1 day. And same principle applies here as well. So a lot of people got together. Usually industry and academia, they work separately. But in this case, for instance, Oxford University originally came up with the vaccine, and AstraZeneca, they joined with Oxford University and used their production engine and their basically their expertise in large-scale manufacturing of drugs, and that's how the vaccine came about. So, I mean, there are, you know, some people are apprehensive about the vaccine because it was produced in such a short time, but, you know, certainly they went through all the necessary tests and you know, regulations, and that's how the vaccines came about.
Sir, there is one more question from Satyashri. Can you share something about CRISPR-Cas9 gene editing tool?
Unfortunately, I've not worked with that one. I can't really comment. That's not really something I've worked on, unfortunately. But that is, you know, that is a really good question. I think CRISPR-Cas9 is one of the emerging, or, you know, in the last 5 years or so, it came about, and that's being used heavily in various aspects of research. About CRISPR-Cas9, you probably need a separate hour for that. That's really complicated, or, you know, a detailed area.
Thank you, sir, for your lucid answers. Gratitude is a quality similar to the electricity. It must be produced and discharged and used up in order to exist at all. I invite Ms. Sanju Nikashni of 2nd MSc Biochemistry to deliver the vote of thanks.
I consider it a great opportunity and privilege to propose a vote of thanks to all the members who made this a memorable and successful event. On behalf of DJYs from our college, I would like to thank our lab secretary, principals, and head of the department who took initiatives to give us a wonderful section. I would also like to thank our speaker, Mr. Vignesh Venkatraman, for spending his valuable time with us for us enlightenments. My hearty thanks to all our faculties who also been there to guide us, support us, and lead us in the right direction towards a successful career. I would also thank our technical supporting team, NCC, and other members of the crew who worked behind the scenes to make this event a great success. Last but not least, I thank all my fellow students and friends for participating in this webinar on recent advance in protein engineering with a focus on antibody drug discovery. Thank you, sir.
Thank you so much for inviting me, and I was, you know, I had a good time talking through the protein engineering and my experience in antibody discovery field. And special thanks to the organizers, the Neuberg crew, and the head of department, Dr. Srinivasan, and also, you know, Nagalakshmi ma'am, for inviting me to give you this talk. So thank you, and all the best to all the students as well. Thank you.